Welcome to our deep dive into the effector functions of immune cells. The immune system is a complex network designed to protect us from disease and infection. At the heart of this system are various immune cells, each with specialized roles. Today we'll explore how these cells identify threats, communicate with each other, and launch targeted attacks to keep us healthy. Understanding these effector functions not only reveals the sophistication of our body's defenses, but also helps us appreciate the delicate balance required for optimal health. Let's start by looking at the main players in the immune system. Immune system is made up of several types of cells, each with unique functions. The primary categories include lymphocytes such as T cells and B cells, as well as innate immune cells like macrophages, neutrophils, and natural killer cells. These cells work together, forming a coordinated defense against pathogens. While some act as sentinels, others are the soldiers on the front lines. In the next scenes, we'll break down the specific effector functions of each cell type and see how they contribute to immune protection. T cells are a cornerstone of adaptive immunity. They come in several forms, including helper T cells, cytotoxic T cells, and regulatory T cells. Helper T cells coordinate immune responses by signaling other cells, while cytotoxic T cells directly kill infected or abnormal cells. Regulatory T cells help maintain balance, preventing excessive immune reactions. These effector functions ensure that the immune response is both effective and controlled, targeting threats without harming healthy tissues. Helper T cells play a vital role in orchestrating the immune response. When they recognize antigens presented by other immune cells, they release signaling molecules called cytokines. These cytokines activate and direct other immune cells, such as B cells and macrophages, enhancing their ability to fight infections. Without helper T cells, the immune system's response would be uncoordinated and much less effective. Cytotoxic T cells are the immune system's specialized killers. They patrol the body, searching for cells infected by viruses or transformed by cancer. Upon recognizing a target, they release toxic molecules that induce cell death, eliminating the threat. This targeted approach minimizes collateral damage to healthy tissues, making cytotoxic T cells essential for precise immune defense. Regulatory T cells are crucial for preventing the immune system from overreacting. They suppress excessive immune responses, reducing the risk of autoimmunity where the body attacks its own tissues. By maintaining this balance, regulatory T cells help ensure that the immune system is both effective and safe, protecting us from disease without causing harm. T cells are another key player in adaptive immunity. Their primary effector function is the production of antibodies. When B cells encounter antigens, they differentiate into plasma cells which secrete large amounts of antibodies. These antibodies bind to pathogens, marking them for destruction and preventing them from infecting cells. B cells also form memory cells, providing long-term protection. Antibodies produced by B cells have several important functions. They neutralize toxins and viruses, block pathogens from entering cells, and tag invaders for destruction by other immune cells. This tagging process, called opsonization, makes it easier for phagocytes to engulf and destroy pathogens. Antibodies are a vital part of the immune system's arsenal, providing both immediate and lasting protection. After an infection or vaccination, some B cells become memory cells. These memory B cells persist in the body for years, ready to respond rapidly if the same pathogen is encountered again. This memory response is the basis for long-lasting immunity and is why vaccines are so effective at preventing disease. Macrophages are large, versatile cells that play a central role in innate immunity. Their main effector function is phagocytosis, where they engulf and digest pathogens, dead cells, and debris. Macrophages also release cytokines to recruit other immune cells and help initiate inflammation, which is essential for fighting infections and healing injuries. Phagocytosis is the process by which macrophages and other phagocytes ingest harmful particles. The cell surrounds the target, engulfs it, and breaks it down using enzymes. This process not only removes pathogens but also clears away dead cells, keeping tissues healthy and free of debris. Macrophages can be activated by signals from other immune cells, such as helper T cells. Once activated, they become more efficient at killing microbes and producing inflammatory molecules. This heightened state allows them to better control infections and support the healing process. Neutrophils are the most abundant type of white blood cell and are among the first to arrive at sites of infection. Their effector functions include phagocytosis, release of antimicrobial molecules, and formation of neutrophil extracellular traps, nets that trap and kill pathogens. Neutrophils are essential for rapid, early defense against bacterial infections. Neutrophil extracellular traps, or nets, are networks of DNA and proteins released by neutrophils to trap and kill microbes. This unique effector function helps contain infections and prevents the spread of pathogens, especially during severe infections. Natural killer NK cells are part of the innate immune system. Their main effector function is to recognize and destroy cells that are infected or have become cancerous. NK cells do this without prior sensitization, making them a rapid and critical line of defense against certain threats. NK cells use a variety of mechanisms to kill target cells. They release cytotoxic granules containing perforin and granzymes which induce cell death. NK cells also produce cytokines that help coordinate the broader immune response, enhancing the body's ability to fight infections and tumors. Dendritic cells act as messengers between the innate and adaptive immune systems. Their main effector function is antigen presentation. They capture antigens from pathogens, process them, and present them to T cells, initiating and shaping the adaptive immune response. Antigen presentation is a critical step in activating T cells. Dendritic cells process antigens and display them on their surface using molecules called MHC. This interaction is essential for T cell activation and the development of a targeted immune response. Mast cells are best known for their role in allergic reactions. When activated, they release histamine and other chemicals that cause inflammation and allergy symptoms. While this response can be uncomfortable, it also helps protect against certain parasites and pathogens. Eosinophils are specialized for combating parasitic infections. They release toxic granules that damage and kill parasites. Eosinophils also play a role in modulating allergic responses and inflammation, highlighting their importance in immune regulation. Basophils are rare but important immune cells. They release histamine and other mediators during allergic reactions and help defend against parasites. Basophils also influence the behavior of other immune cells, contributing to the overall immune response. Effector cells are activated immune cells that carry out immediate defense functions, such as killing pathogens or producing antibodies. Memory cells, on the other hand, persist long-term and respond rapidly upon re-exposure to the same pathogen. This division of labor ensures both immediate and lasting protection. Cytokines are small proteins released by immune cells to communicate and coordinate responses. They can stimulate cell growth, attract other immune cells, and regulate inflammation. Cytokines are essential for ensuring that the immune system acts efficiently and appropriately. Chemokines are a special type of cytokine that guide the movement of immune cells to sites of infection or injury. By creating chemical gradients, chemokines ensure that immune cells arrive where they are needed most, enhancing the effectiveness of the immune response. The complement system is a group of proteins that work with immune cells to destroy pathogens. Complement proteins can directly kill microbes, enhance phagocytosis, and promote inflammation. This system acts as a powerful amplifier of immune responses. Inflammation is a hallmark of immune activation. While it helps contain and eliminate infections, excessive inflammation can damage tissues. Effector immune cells carefully regulate inflammation to balance defense and tissue protection, highlighting the importance of immune control. Immune memory is the ability of the immune system to remember past infections. Memory cells, both T and B cells, provide rapid and robust responses upon re-exposure to the same pathogen. This principle underlies the effectiveness of vaccines and long-term immunity. Antigen recognition is the first step in immune activation. Immune cells use specialized receptors to detect foreign molecules called antigens. This recognition triggers a cascade of effector functions, leading to the elimination of the threat. Opsonization is the process by which antibodies and complement proteins tag pathogens for destruction. This tagging makes it easier for phagocytes to recognize and engulf invaders, streamlining the immune response and increasing efficiency. Antibody-dependent cell-mediated cytotoxicity (ADCC) is a process where immune cells such as NK cells recognize antibody-coded targets and destroy them. This mechanism bridges innate and adaptive immunity, providing a powerful means to eliminate infected or abnormal cells. Immune surveillance is the ongoing process by which immune cells monitor the body for signs of infection or abnormality, such as cancer. This constant vigilance allows for early detection and rapid response to threats, keeping us healthy. Immune tolerance is the ability of the immune system to distinguish between self and non-self. Regulatory T cells and other mechanisms prevent attacks on the body's own tissues, reducing the risk of autoimmune diseases and maintaining health. Autoimmunity occurs when the immune system mistakenly attacks the body's own cells. This breakdown in tolerance can lead to chronic diseases. Understanding effector functions helps researchers develop therapies to restore balance and prevent tissue damage. Immunodeficiency is a state where the immune system's effector functions are impaired, making the body more susceptible to infections. This can be inherited or acquired, as in HIV infection. Understanding effector mechanisms is key to developing treatments for these conditions. Vaccines work by stimulating the immune system to develop memory cells and effector functions without causing disease. This prepares the body to respond quickly and effectively if exposed to the real pathogen in the future. Immunotherapy harnesses the power of immune effector functions to treat diseases like cancer. By boosting or redirecting immune responses, these therapies offer new hope for patients and highlight the importance of understanding immune mechanisms. Immune checkpoints are regulatory pathways that control the intensity of immune responses. Cancer cells can exploit these checkpoints to evade immune attack. Therapies that block checkpoints can unleash effector cells against tumors, revolutionizing cancer treatment. Cytotoxic granules are specialized structures in cells like cytotoxic T cells and NK cells. They contain molecules such as perforin and granzymes that induce death in target cells. This mechanism is crucial for eliminating infected or cancerous cells. Granulocytes are a group of white blood cells that include neutrophils, eosinophils, and basophils. Their effector functions involve releasing granules filled with enzymes and toxic substances to combat pathogens and regulate inflammation. Effector functions are rarely carried out by a single cell type. Instead, immune cells cooperate, sharing information and supporting each other. This teamwork ensures a coordinated and effective defense against a wide range of threats. Communication between immune cells is essential for mounting an effective response. Cells use cytokines, chemokines, and direct contact to relay information, adjust strategies, and coordinate their actions. This intricate network of signals keeps the immune system responsive and adaptable. After a threat is eliminated, the immune system must resolve the response to prevent unnecessary damage. Effector cells either die off or become memory cells, and regulatory mechanisms suppress inflammation. This resolution phase is vital for restoring balance and tissue health. Effector immune cells not only fight infections but also support tissue repair. Macrophages and other cells release growth factors that promote healing and regeneration. This dual role highlights the importance of immune cells in both defense and recovery. Disorders of the immune system can arise when effector functions are too weak, too strong, or misdirected. These include immunodeficiencies, autoimmune diseases, and allergies. Understanding effector mechanisms is key to diagnosing and treating these conditions. Effector functions of immune cells are central to our body's defense against disease. From direct killing to communication and memory, these functions ensure both immediate and long-term protection. Advances in immunology continue to reveal new insights, offering hope for better treatments and healthier lives. Thank you for joining us on this journey through the remarkable world of immune cell effector functions.